This video is to go over the core concepts of the central dogma. So here's what I would suggest that you be able to recapitulate for an exam. I would encourage you to have this kind of short list so you can write it down right away when you get your exam and then refer to it throughout the questions. Basically, we start off with the basics, DNA to RNA to protein. And of course, this is actually messenger RNA that's used as the instructions to make protein. And then we can add in some more details, for example, what the name of each process is. So using a DNA template to make messenger RNA is the process of transcription, whereas using messenger RNA as a template to make protein is translation, or TLN. Now we can add in the names of the enzymes that are used for each of these. RNA polymerase is the core enzyme that is used for transcription, and like its name implies, it is used to make a polymer of RNA. Then during the process of translation, the enzyme is the ribosome. And as you know, we start with DNA, which is double-stranded, then we make messenger RNA, which is single-stranded, and then finally we make a protein, which is often shown as a blob, but really is a string of amino acids that are covalently bonded to each other. And let's write in a couple of the things we want to keep track of. So for messenger RNA, some of the important things are to keep track of which side is the 5' versus the 3'. And of course for DNA, we've got two strands, each of which has a 5' and 3', and they run anti-parallel to each other. Then for the protein, we have an N terminus, and then we have a C terminus, and that corresponds to which functional group is protruding from that side. The amino group is protruding on the N terminus, whereas the carboxyl group is protruding from the C terminus. Now I want to go into some more details about each of these processes, so let's draw that below here. So a lot of times our attention is directed at the details of transcription, and so here I'm drawing the DNA double helix, and then let's consider how that's being used as a template, and that we have our protein blob, which is RNA polymerase, which is reading the DNA. And so now we can add in some more details here. Remember that RNA polymerase adds only to the 3' end of the growing RNA strand, which means this is the 5' end over here, and this is 3' here. When we label the DNA strands, remember that we have different names for them. We have the template strand, and then we have the coding strand. And the template strand is the strand that RNA polymerase is physically interacting with and is reading. So that's our template strand. And that's going to run opposite direction, 3' to 5'. And then we have the coding strand up here. And so that's going to run the same direction as the messenger RNA, which is 5' to 3'. As an example, let's consider if we have AUG here. Remember that on the template strand, then that would mean that we would have TAC. And when we consider what's on the coding strand, then we would have A, T, and G. So that's corresponding to those nucleotides. Now let's add in a drawing for what translation looks like. Most of our attention has been focused at elongation when we have the small subunit and the large subunit. And we've got our 5' and 3' of our messenger RNA here. We have our AUG in this position. Then we have our transfer RNA here. And we could add in a bunch of labels for this, like the small subunit of the ribosome. And the large subunit. We've got a transfer RNA here. And the little circle is amino acid. Now, this is not exactly the same order that we talked about how these come together for initiation, so you can watch the other video for that. But just to add in a few more of the details, we could have another transfer RNA here, and then we have this site here for the site of peptide bond formation, which again occurs between these two adjacent amino acids, and this would be cleaved. Okay, so what I was saying about what I would encourage you to have handy to jot down during the exam, I would basically stick with this stuff here, DNA, messenger RNA to protein, write down these core names of the processes and their enzymes, then have a quick drawing here, probably not add in like all of these names, but basically at least have the drawing of what's shown in blue so you could have kind of a visual for what the process is. The reason why I'm telling you to do that is that, especially for the multiple choice section, you're going to see options for both of these processes, but I'll be asking you about one of them. And so it's going to be distracting to see all of those things. If you have a little reference that you can use, which you have kind of you've committed to memory and then can write down on the exam, then you can do a quick reference to that as you go through those questions and pick up as many points as possible. Okay, so now let's talk a little bit about some of the things that make these processes different in prokaryotes and eukaryotes. So for prokaryotes versus eukaryotes, some of the things that are different include transcription initiation, and this includes things like the -10 and -35 DNA sequences, which are the promoter, and then for eukaryotes, the Tata box, which again is made of DNA, and it is the promoter. Each of these processes and the respective organisms also has a protein with it, and so for prokaryotes, that's sigma factor, which is a protein, and it binds to the promoter. Then at the Tata box, they are called basal transcription factors. Those are proteins, and again, they bind to the promoter. And this combination of protein at the promoter will help recruit RNA polymerase to come to that location for that particular gene and initiate transcription. Same thing over here, promoter plus the respective proteins encourages RNA polymerase to come to that position and to start transcription. Now you should also know some things about transcription elongation. This is basically the same thing for both prokaryotes and eukaryotes. Basically, we use RNA polymerase. RNA polymerase finds the complementary nucleotide to whatever nucleotide that it is reading on the template strand. And of course, we use A, U, C, and G for RNA, which corresponds to A, T, C, and G as complementary nucleotides on the DNA. And the big thing here is to know that the RNA polymerase enzyme always adds to the 3' end. So it's basically the same process for prokaryotes and eukaryotes. And then let's talk a little bit about transcription termination. So RNA polymerase does not use all of the DNA in the genome, it just uses a very small portion of it. And so basically it needs to have these termination cues. And for prokaryotes, this is the hairpin loop. Which makes this sort of shape, and this is made of RNA. So this is part of the messenger RNA that, as it's transcribed, will form this three-dimensional structure. RNA polymerase does not like this extra shape. It starts to annoy it or destabilize it. It's more accurate, and then RNA polymerase leaves. That's for prokaryotes. For eukaryotes, for transcription termination, instead, this is the addition of the poly A tail, which again is extra stuff that gets added, and this also destabilizes RNA polymerase. Okay, now let's talk a little bit about the translation, 3 steps: initiation, elongation, and termination, and how they are different. So for translation, for initiation, this is different for prokaryotes versus eukaryotes. Basically for prokaryotes, there is a ribosome binding site. It is in this little region here and includes the AUG. You don't need to know all these details, that's a little bit more than the AUG, and this is what the small subunit of the ribosome recognizes and binds to. So the ribosome binding site, this is part of the messenger RNA, and it attracts the first subunit of the ribosome, the small subunit. Then for eukaryotes, instead it's the 5' cap, which again is on the RNA, and this also attracts the small subunit of the ribosome in these organisms. Now let's talk about translation elongation. Basically, this is the process of new transfer RNAs coming into the A site of the ribosome, and this is going to be transfer RNAs that correspond to the codon that is exposed in this location. And basically, we're going to have a peptide bond form in between those adjacent amino acids based on the catalytic ability of the ribosome in that position. And so basically, this is a peptide bond formation. That's my dog, Kona. Hello. So peptide bond formation between adjacent amino acids. And the N-terminus is always going to be on this side. That's the first amino acid that's added. That's always called the N-terminus. And then if we were going to stop our translation after this second amino acid, this would be the C-terminus. That's always the most recent amino acid that's added. And again, that has to do with bond chemistry between the amino group and the carboxyl group in this position here. Carboxyl group on this guy and amino group on this guy. And this happens the same way in prokaryotes and eukaryotes. Then let's talk about translation termination. It's tempting to think that this is just the stop codon, but it's more than that. So translation termination is when a stop codon is unveiled in the A site. This would not be the case here because we see a transfer RNA. Instead, for translation termination, a release factor, which is a protein that corresponds to a stop codon, goes into the A site of the ribosome. The ribosome, kind of like RNA polymerase, is a big enzyme. It has a lot of things to do. It gets annoyed when things fit into places that it would normally be doing its catalytic activity. And so the large and small subunit of the ribosome will break apart from the messenger RNA. They will cleave the last amino acid off of the transfer RNA, and then that will release the newly made polypeptide, which can then go through protein folding. Okay, now I want to add just a couple more little details to help you kind of see how things fit together. So something else we talked about at the very beginning of this unit was steroid hormone signaling. And what I want you to know is that the steroid hormone signaling, as you may recall, can tie into information in the nucleus and turning genes on and off. And so basically what steroid hormone signaling does is kick off the process of transcription and then eventually translation. And so this is all part of our larger flow of information within the cell. And then just to kind of wrap that up, remember that proteins, they can be released, and these can, or they can stay in the cell, and these can change behavior or function of the organism.